said that it was from when the cousin smashed so hard. Once there was this girl who wouldn't go and change with the girls in the change room, and when they finally made her, they saw birthmarks all over her body. She couldn't quite explain it, they'd always just been there. But both girl and boy were glad the one kid had it worse than that. Instead, there was this boy whose parents made him come directly home right after school, and when they went to their church, they shook and lurched all over the church floor. You couldn't quite explain it, they'd always just gone there.
Mengumumkan keberangkatan masuk Duli Yang Teramat Mulia Raja Muda Perak Darul Ridwan, Raja Dr. Nazrin Shah, Tuanku Pro-Chancellor University Malaya, diiringi oleh pengurusi Lembaga Pengarah University dan Naib Chancellor University Malaya, Dev Kehormat. Menghadap Duli Yang Teramat Mulia Raja Muda Perak Darul Ridwan, Raja Dr. Nazrin Shah, Tuanku Pro-Chancellor University Malaya. Ampun Tuanku, beribu-ribu ampun. Sembah patik pohon diampun. Bagi pihak hadirin sekalian, menjunjung setinggi-tinggi kasih di atas perkenan tuanku. Pro-Chancellor berangkat mendirikan majlis pada pagi yang indah ini. Untuk meneruskan acara pada pagi ini, patut memohon perkenan tuanku untuk berucap dalam bahasa Inggeris. Ampun tuanku. Yang berbahagia, Tan Sri Dato' Arsyad Ayub, Chairman, Board of Directors, University of Malaya. Professor Gauss Jasmond, Vice Chancellor, University of Malaya. Mr. Uwe Morawetz, Founding Chairman of the International Peace Foundation. Yang berbahagia, Dato' Insinyur Dr. Haji Ahmad Zaidi Laidin, Vice President of the Academy of Sciences, Malaysia. The distinguished Professor Howard Robert Horvitz. Ladies and gentlemen, thank you for being here this morning. I wish to extend a warm and cordial welcome to everybody present at this unique event today. This morning promises to be a rare intellectual feast. We look forward to it with great anticipation. This event is made possible by the gracious presence of Nobel laureate Professor Howard Robert Horvitz, fondly called Bob by family members and friends, through the collaborative effort of the University of Malaya with the International Peace Foundation. Ladies and gentlemen, we in this university are fortunate enough to have the leadership of a Vice Chancellor who has single-mindedly pursued a clear and unrelenting course towards greatness. With unceasing energy and bold enthusiasm, he has inspired and enthused the rest of us. We are therefore proud to welcome to the stage Professor Gauss Jasmond, Vice Chancellor, University of Malaya, to deliver his address.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi ta'ala wabarakatuh dan salam sejahtera. Menghadap duli yang teramat mulia, Raja Muda Perak Darul Ridwan, Raja Dr. Nazrin Shah, Ibni Sultan Azlan Muhibbuddin Shah, Tuanku Pro-Chancellor Universiti Malaya. Ampun Tuanku. Patut sekalian memanjat setinggi-tinggi kesyukuran kehadrat ilahi kerana dengan limpah izinnya, Tuanku sehat walafiat dan dapat berangkat untuk bersama-sama patut untuk mendengar syarahan oleh Profesor Howard Robert Horvitz pada pagi ini. Patut menjunjung setinggi-tinggi kasih di atas perkenan Tuanku mencemaduli bagi menjayakan syarahan ini. Seterusnya patut mohon perkenan Tuanku untuk menyampaikan sepatah dua kata kepada para hadirin sekalian dan untuk patut berucap dalam bahasa Inggeris. Ampun Tuanku. Mr. Uwe Morawetz, Founding Chairman of the International Peace Foundation, our distinguished speaker, Professor Howard Robert Horvitz, members of the Academy of Sciences Malaysia, professors and academics of the university, distinguished guests, ladies and gentlemen. The University of Malaya is very honored to host this public lecture by Professor Horvitz entitled Basic Biomedical Research Drives the Discovery of New Treatments for Human Diseases. The lecture is a collaboration under the auspices of the Bridges series, in which Mr. Uwe Morawetz will speak more about this in a short while. Ladies and gentlemen, what I would like to say is how apt and succinct the topic of Professor Horvitz is. Whilst he will focus on biomedicine, the overarching essence is the importance of basic scientific research. The horizons of knowledge, be it in the sciences or the humanities, are built upon the building blocks of basic research. Basic research looks at the fundamentals of life, space, and the universe, spanning various fields such as physics, chemistry, mathematics, biology, anthropology, archaeology, sociology, culture, and so on. How man evolves through time, how humanity impacts upon life on earth, how technology affects the quality of life of man, how language and culture color the civilizations of man through the ages, and the interaction with each other. For basic research to thrive, it requires passion, a fieriness of spirit, and an immersion of researchers in the disciplines. These researchers are sometimes the unsung heroes. They slog to unravel the mystery and the magic of life at the most basic and micro level. When we understand why some things behave the way they do, or why they react the way they do in certain conditions, or how we can tweak them to behave the way we want them to, then we can proceed to harness their various benefits through technological innovations. For universities like the University of Malaya, basic scientific research is the cornerstone of all other research activities. These activities take time to flourish and bear results. As such, the researchers need funds, space, and equipment, research assistants, and other auxiliary staff, as well as linkages with other top brains around the world. One such top brain is today here to address the academic community of the university. I am very happy and thankful that Professor Horvitz has chosen to speak on how basic biomedical research contributes towards the treatment of diseases for mankind. The University of Malaya has a steep target of attempting to bring itself to be within the top 100 universities in the world within 5 years. Your visit here today, sir, will be a source of great inspiration to all our academics to work hard and produce quality research outputs which will serve to push our credentials higher. We have a fairly large number of biomedical researchers in the campus, and they will certainly be inspired by your talk today. I wish to thank the International Peace Foundation and all the sponsors for making it possible for Professor Horvitz to share his wealth of knowledge and experience with us today. I also thank everyone for making time to attend this very important talk. We have guests from the corporate sector, the universities, colleges, schools, and members of the public. Above all, I would like to record our deepest appreciation to His Royal Highness the Crown Prince of Perak for consenting to grace the lecture. Ampun tuanku, Patik sekali lagi menjunjung setinggi-tinggi kasih di atas lempah perkenan tuanku berangkat pada pagi ini. Ampun tuanku. Sekian, wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Mr. Vice Chancellor. This series is made possible by the collaborative initiative of the University with the International Peace Foundation. Let's welcome Mr. Uwe Morawetz, founding chairman of the International Peace Foundation, to deliver his speech.
Your Royal Highness, Excellencies, ladies and gentlemen, friends, welcome to the second ASEAN event series, Bridges, Dialogues Towards a Culture of Peace. Bridges is facilitated by the International Peace Foundation, a non-political and non-religious foundation under the patronage of 21 Nobel Peace Prize laureates based in Vienna. The events are hosted in cooperation with various local partners, including the country's major universities, and I thank the University of Malaya and its Vice Chancellor, Professor Gaut Jasmon, for hosting our event today. I'm grateful to the Malaysian Honorary Chairman of Bridges, His Royal Highness Raja Nasrin Shah, and to our Chairman, Tun Musa Hitam, Their powerful guidance paved the way for Bridges to bear fruit. Since November 2008, Bridges is being continuously held in Malaysia and Thailand until April 2009, involving the participation of Nobel laureates and other eminent keynote speakers. The second ASEAN series of Bridges is an independent contribution to the Decade for Culture of Peace and Nonviolence, initiated by the United Nations General Assembly. It follows the series of 300 Bridges events, which the International Peace Foundation has already hosted in Thailand and the Philippines since 2003. 30 Nobel laureates, as well as 13 other keynote speakers and artists such as Dr. Hans Blix, Dame Anita Roddick, the Reverend Jesse Jackson, Vanessa May, Jesse Norman and Dr. James Wolfenson participated in these events. In Thailand, they were presided over by Her Majesty Queen Sirikit and Her Royal Highness Crown Princess Maha Chakrisirinthorn, and reached an audience of 90,000 participants. As peace cannot be achieved instantly, but is a process which needs time, Bridges has not been organized as a single conference, but as an ongoing series of events in which Nobel laureates and international decision makers have built strong bridges with leaders in all parts of society and with the general public. With the basis for peace being education and synergies being the fruit of cooperation, the International Peace Foundation has not realized Bridges alone, but has carried out the program together with UNESCO and 88 other national and international institutions, including 33 major universities and schools. The multidisciplinary and pluralistic approach of Bridges in Thailand and the Philippines and now in Malaysia reflects that peace involves all parts of society. It involves awareness and social responsibility of politicians, the business community, scientists, artists, and the media. And since peace within ourselves, our families, and our environment starts in our minds and hearts, it involves every one of us. In this sense, Bridges challenges us to cross borders and to build bridges by listening and opening up to other viewpoints, by generating new thoughts, by developing innovative forms of cooperation, and by fulfilling the desire of everyone to get to know and to learn from each other. This can lead us to a world in which we will be able to understand each other and the complexities we face today in a new light. A globalized world needs broad strategies for change to secure a sustainable future for us and the next generations. Let us be inspired by the knowledge and the wisdom that Bridges continues to offer. An opportunity to get a more inclusive, interconnected, and comprehensive view of ourselves and the world in which we live in which we are able to create anew constantly, through dialogues towards a culture of peace which need the participation of everyone. I thank Professor H. Robert Horvitz, the 2002 Nobel Laureate for Medicine, who has agreed to come to Malaysia to support the events. We now look forward to his keynote speech and to his important contribution to build bridges. Welcome, Professor Robert Horvitz.
First, I would like to thank Mr. Uwe Morawetz and the International Peace Foundation for inviting me to participate in this very important program. The words of inspiration that we have just heard are, to me, very moving, and I hope that all of us will share in their content, meaning, and future hope. I'm very honored to be here, and I'm very honored to have been included in the exceptional group of visitors who have been hosted by this program to date. I'm also exceedingly honored for His Royal Highness to be here today and help host me and have very nice conversation with me already this morning. Personally, I am appreciative of the opportunity for me and my family, my wife and daughter, to visit Malaysia for the first time. We have heard much about Malaysia. We've had 2 young women live with us over the past 4 years from Malaysia. So I feel like I know something about the country, but the reality of being here is very exciting and something that means a lot to me. Now, I've entitled my lecture today, Basic Biomedical Research Drives the Discovery of New Treatments for Human Disease. And in this lecture, I will exemplify how basic research in biomedical science can lead to discoveries that promise to provide new approaches to disease. This lecture, as you know, is hosted by the International Peace Foundation, so I will begin with a few comments about how I see biomedical science, world health, and world peace all vitally interconnected. Specifically, I believe that there are many threats to world peace around the world, and one of the major threats is inequalities amongst people. There are inequalities in wealth. There are inequalities in education. And there are inequalities in health. And health is really the most fundamental of these. Without good health, neither the education nor the wealth of a society can really be improved. And I think about this in the context of a conversation I had a few years ago with Daniel Vasella, who is the CEO of the international pharmaceutical giant Novartis. And Dan was sitting next to me at dinner and telling me that he had established a foundation to try to address this issue of inequalities amongst peoples around the world. The first project of the foundation was the building of a school in Mali, in West Africa. And the school had been built, and Dan went from Switzerland to Mali for the dedication of this school. And what he found was very disheartening. What he found was that the school was likely to have little impact. And the reason was the children simply, in this village in Mali, were too ill to take advantage of the opportunities offered by this new school. And so what Dan learned from this is that even before education, and of course education is vital, must come health. And so with his knowledge of the pharmaceutical industry and his knowledge of global health, he has turned his interest now first to addressing the problem of health. And it is within this context that I want to describe to you my thoughts about this area. So how can we collectively begin to think about the problem of improving global health? And I think there are 2 answers, at least, to this question. First, there must be broad international consortia that focus on global health. And such efforts must involve the active participation of governments, of foundations, of pharmaceutical companies, of hospitals and other groups involved in healthcare, and probably most importantly, must involve very, very dedicated individuals, because it is really people who can drive change and progress. And each of us has a responsibility to do the best we can in this sort of area. There obviously must be appropriate financial support, and each country must develop the infrastructure to implement changes in healthcare to try to optimize the health available to its populations. So that's one side of a coin. The other side is there must be advances in the diagnosing, treating, and preventing of disease. Many of today's diseases we don't know how to treat. In some cases, the treatments are not very good. In some cases, the treatments actually have such harmful effects that they are dangerous to give. And in some cases, the treatments are so expensive that they cannot be considered for use in any kind of a global context. Furthermore, sometimes resistance arises to existing treatments, and that requires the development of new treatments, sometimes progressively, treatment after treatment. So that, for example, here we know about malaria, and the fact that much malaria, probably most of the malaria in the country, is resistant to the drug chloroquine, which was a very good drug, but no longer is useful. So we have now new drugs, and most likely we're going to need new drugs beyond that. Now, where do new methods of diagnosing, treating, and curing disease come from? And my thesis today is that such advances are crucially dependent on discoveries made and driven by basic biomedical science. And in this lecture, I will demonstrate how basic research in biology can lead to discoveries that promise to provide new approaches to the treatment of disease. I will also advocate that both governments and foundations should allocate a significant portion of their research portfolios to basic research, even though countries may be faced with very pressing specific needs for applications of research. Applied research, I will argue, cannot be done effectively if there isn't a very strong base of basic research to support and promote it. Now, today I'm going to focus on research from my laboratory, research that resulted in my receiving, my sharing really, with two others you'll hear about, the Nobel Prize in Physiology or Medicine in 2002. Now, many people know that the Nobel Prize comes with a gold medal. People also know that the Nobel Prize comes with some money. And both of these are very nice, but what I didn't know is that the Nobel Prize also comes with a diploma. This slide shows my Nobel diploma. And if you read it at the end, assuming you can all read Swedish, you'll see that in part what it says is that this prize was awarded for understanding the genetic control of programmed cell death. So what does this mean? Why did I receive a Nobel Prize for studying the genetic control of programmed cell death? Let me back up to some of the words. Genetics, cell, programmed cell death. Genetics. Genetics is the study of genes. Genes are responsible for all of the biological processes that occur in living organisms. Genes are the basis of heredity. Each of us has inherited half of our genes from our father and half of our genes from our mother. Genes can vary amongst individuals, so we are all genetically distinct, but we are genetically more similar to each other than we are to other species. Variations in genes can also cause or predispose us to disease. And many, many diseases are diseases caused by abnormalities in genes, and many other diseases we are more susceptible to, more likely to get if we carry specific genetic variants. And these diseases include cancer, cardiovascular disorders, asthma, cystic fibrosis, premature aging, Alzheimer's disease, bone loss, and I could go on with diseases that are caused by or influenced by our genes. So genes are important to us, and they are crucial to our health. So how can we learn about our genes? How can we learn what they do and how they sometimes go wrong? Now, one approach is to study our genes, human genes. And many biologists and biomedical researchers do precisely this. I do this. I study human genes that are responsible for the neurodegenerative disease amyotrophic lateral sclerosis, or ALS. But there's a difficulty in studying human genes. Such studies in many ways are slow and inefficient, and there are many types of studies that you simply cannot do with people. So one wants to be able to study genes, including human genes, in some more rapid, analytical fashion than can be done easily with our own genes. Fortunately, biology has provided us with an approach to solving this problem. And that's based upon the fact that many genes are strikingly conserved amongst different organisms. And what this means is that we can study genes in experimental organisms, easily studied organisms, learn what they do in these organisms, and then extrapolate from that information to what they do in us, and then test the hypotheses to determine if in fact this is true. So many experimental organisms are used in modern biomedical research. For example, to study genes, people will study mice, or zebrafish, or fruit flies, or yeast, single-celled organisms that are used to brew beer and bake bread. Another organism that is commonly used today in genetic studies is a microscopic roundworm, a nematode. It's called Caenorhabditis elegans, or C. elegans for short, and this is the organism that I have studied. And I'll turn back to C. elegans in a few moments and tell you a little about it. So those are some words about genetics. Now, what about cell? What is a cell? In short, a cell is the fundamental unit of life. Our bodies are made up of cells, skin cells, muscle cells, blood cells, nerve cells, and so on. We have genes, we have cells. What about programmed cell death? Programmed cell death refers to naturally occurring cell death, i.e. cell death that occurs as a normal part of the genetic program of every animal. And let me explain in more detail what I mean by that. When an animal develops, such as us, we begin as a fertilized egg. Sperm and egg come together. That makes a single cell. It divides to make two. Those two divide to make four, and so on over and over again until many cells are present. In us, as many as 10 million cells in each of us. Then these cells take on specific characteristics, becoming nerve cells, muscle cells, blood cells, and so on. And the cells interact to form structures like hands and a nose, and in a more complicated way, a brain. These processes of cell division, cell differentiation, and morphogenesis are the fundamental processes of development, of animal development, and of the field of developmental biology. Now, in addition to these fundamental processes, there is another process that is universal amongst animals, and that is the process of cell death. Quite remarkably, and I would say rather counterintuitively, many of the cells that are generated as animals develop do not survive, but rather die. And it's this naturally occurring cell death that is referred to as programmed cell death. The phenomenon of programmed cell death has been known for many years. For example, if you think about a tadpole as it metamorphoses into a frog, the tadpole loses its tail. The tail is made of cells, and those cells die. That is programmed cell death. If you consider a human infant in utero, when a baby is developing, between its digits, its fingers and its toes, there is webbing. The webbing is made of cells, and that webbing is removed before birth by the process of programmed cell death. If you think about birds, particularly bird feet, some are webbed, some are not. The ones that are not, there has been programmed cell death. The ones that are webbed, there is no programmed cell death. Programmed cell death can be a major event. As our brains develop, as many as 85% of the nerve cells that are generated die. As we sit here today, in our blood, many of the blood cells, 95% of a particular kind of blood cell known as a thymocyte, 95% of the thymocytes generated will die by programmed cell death. So programmed cell death is widespread in biology. Yet, until a few years ago, biologists paid little attention to this phenomenon. And the reason, I think, was that when one thinks about the challenges of life, how do you make life? That seemed like a big problem. And the default, I think, was if it didn't work right, then you get death. So dying cells were cells that had been mistreated, cells that had been harmed, cells that had not been supplied with appropriate nutrients, and not interesting because that was a default. What I and my colleagues discovered was that this view is incorrect. Rather, there is a biology of cell death every bit as much as there is a biology of other fundamental biological processes like cell division and cell differentiation and morphogenesis. And that there are specific genes that act to make cells die. So I've been talking now about cell death and the biology of cell death, and in fact, any biology, any human biology, if it goes wrong in us, can lead to disease. And it turns out that scientists have been finding in recent years that programmed cell death, now that we're thinking about it, is no exception to this. Many diseases are diseases of a misregulation of programmed cell death. There are diseases in which there is too much cell death. For example, neurodegenerative diseases in which certain nerve cells die. Alzheimer's disease, Huntington's disease, Parkinson's disease. Notably, certain retinal diseases which cause blindness. Stroke, brain cells die. Traumatic brain injury, aspects of AIDS. Heart cells die in heart attacks and congestive heart failure. Many liver diseases, including infection with hepatitis C virus, involves deaths of liver cells. And it turns out that these liver cells are dying using the same genes that are normally used by us when we develop. These genes have been unleashed at the wrong time or in the wrong cell, causing these cells to die. On the other hand, there are diseases in which there is too little cell death, certain autoimmune diseases, and also cancer. We think about cancer as coming from cells dividing and dividing and dividing uncontrollably, generating too many cells. But what is happening in our bodies is that many tissues have cells in a number that is defined by two opposing processes. Cells are being added by cell division, and cells are being taken away by cell death. And we can get too many cells either because there is too much cell division or because there is too little cell death. And certain cancers are more fundamentally diseases of too little programmed cell death than of cell division per se, like follicular lymphoma exemplified in this slide. And in fact, all cancers seem to involve too little cell death. And when we treat cancers with radiation or with chemotherapy, what we are doing is activating that endogenous program for programmed cell death and making our cells express this program that is used normally developmentally to cause cells to die. So it was our discoveries about the understanding of the process of programmed cell death, in fact, that led to the Nobel Prize. And we analyzed this process not by studying it in humans, but rather by studying this microscopic worm, 1 millimeter long, so tiny you can barely see it with the naked eye, this microscopic worm, C. elegans. 3 of us shared the prize. My co-recipients were Sidney Brenner and John Sulston. Sidney introduced C. elegans to the field of modern biology, and he worked out the basic methods for the study of this organism. He chose the organism because it is very small and very simple and very easily studied. John Sulston worked with Sidney in Cambridge, England, in the laboratory of molecular biology from the Medical Research Council. And John studied the developmental biology of C. elegans, and in particular, he studied that pattern of cell divisions that occurs beginning with the single-celled fertilized egg and ultimately generating the adult animal. And John studied the complete pattern of this so-called cell lineage. C. elegans is the only animal for which we know the origin of every cell in the animal. And as John studied this process, he discovered that the animal generates more cells than is found in the adult. The adult has 959 cells, much simpler than the 10 million million in us. The adult also has lost 131 cells that during the course of development have undergone programmed cell death. What I and my laboratory did was then to study the mechanistic basis of the programmed cell deaths that John Sulston identified as occurring. And the rationale was, if we could understand the mechanism of programmed cell death in C. elegans, we might get some clues about how cell death occurs in us, and then how it goes wrong in this vast array of diseases that involve abnormalities in programmed cell death. So what we did was to seek genetic variants of C. elegans that were abnormal in programmed cell death. For example, in the top panel of this slide, you can see where the arrows are pointing a number of cells that are undergoing programmed cell death, and in the lower panel is a mutant in which these programmed cell deaths do not occur. This mutant is abnormal in a gene we name SED3, CED for cell death abnormal. And when SED3 is inactivated by a change in its DNA, cell death does not occur. Turn off SED3, no cell death. What does SED3 do? In short, SED3 is a killer gene. It makes cells die by programmed cell death. And it was this discovery that said there is a biology of cell death. There are genes that are acting actively to make cells die. We identified a second killer gene. We named it SED4. We studied these two genes to ask the question, where do they work in the animal? And the answer was they work within those cells that are going to die. So they are suicide genes. They are causing cells to die by acting within those cells. We identified a third gene that acts oppositely, a gene called SED9. SED9 is a protector gene. It prevents cells from dying. And then we identified a fourth gene called EGL1, which is another killer. So we had three killers and a protector. And we analyzed them and determined how they interact. And it turns out that SED3 directly kills. SED4, the killer, works by activating SED3. SED9 protects by preventing SED4 from activating SED3. And EGL1 kills by preventing SED9 from preventing SED4 from activating SED3. We have a pathway, and we understand basic order of action. We analyzed the genes further, and in short, what we found was that each of these genes has a human counterpart. For example, if we look at the gene SED9, a protector gene, the human counterpart is a gene called BCL2. That's a human cancer gene. That is the gene that protects cells from dying, and in cancer protects cells, B cells, in our blood from dying, leading to a proliferation of B cells and the cancer follicular lymphoma. Okay. More generally, each of the four genes has a human counterpart, and it turns out they interact in precisely the same way that we described for the worm. So our pathway derived from studies of this tiny worm has led us now to an understanding of the genetic and molecular control of this process of programmed cell death in people. So what can be done with this information? Well, now we think about treating human disease. And you'll remember that there are two kinds of diseases in which programmed cell death is crucially involved. One, there are diseases with too much programmed cell death. What can we do about such diseases? We can inhibit a killer. So, for example, the gene SED3 is a member of a family today known as caspases. These are killer genes. And if we can develop drugs to inhibit these killer genes, then we could block these diseases that have too much cell death, like neurodegenerative disorders, cardiovascular diseases, liver diseases, and so on. On the other hand, there are diseases like cancer, where there's too little cell death. What do we want to do then? We want to inhibit the protectors, block the protection, activate the cell death, and kill those cells that should normally be dying but in the disease are not. And so what we do then is we develop molecules that can inhibit the protectors, and that gives us molecules that are potential therapeutics for cancer and autoimmune disorders. Now, when my laboratory first made these discoveries, I founded a biotechnology company based upon the science of programmed cell death. And this company has developed 2 drugs. And these drugs now are both in the hands of major pharmaceutical companies. My little company no longer exists. It was acquired by another company. And these 2 drugs are now in the hands of major pharmaceutical companies, which are developing them on the one hand for cancer and on the other hand for liver disease. And I'm very hopeful that these drugs, as well as drugs that are being developed more broadly in the biotechnology and pharmaceutical industries, will be useful very broadly to treat human disease. Now at this point, I'd like to put the discoveries that I've described into a broader context and really address the title and theme of today's lecture. The work I've described involved absolutely basic research. When I began, neither the generality nor the application of our efforts was at all clear. The organism I worked with, this roundworm, this microscopic worm, was obscure even to biologists. In fact, I was advised not to work on it because my career would end if I worked on this obscure animal. Genetic studies are often very abstract. I did not target any disease. When I began, we didn't know if we would be working on anything of any general relevance. In fact, I didn't even know if what we found really would prove to be important to any organism other than this worm. Nonetheless, our studies established mechanisms that appear to be universal amongst animals, and our findings, I think, are likely to help provide the basis for new treatments for a broad variety of human diseases. So I think there are a number of important messages here. First, basic research, and by that I mean research that is discovery-based, will very often lead not only to intellectually interesting findings, and I think intellectually interesting findings are very important, but there's more, and that is that such basic research will also lead to the insights, to the innovation that will be the driver of future practical knowledge. Basic research, biomedical and other, is the driver of new knowledge. Okay? Now, how and where should basic research be supported? I've worked extensively with biotechnology, pharmaceutical, and venture capital companies. And I can tell you that basic research cannot really be supported by the private sector. If you don't know ahead of time the area in which basic research is going to lead to discovery, you cannot develop a viable business plan for a company, not for true basic research. For this reason, I strongly advocate that governments and foundations must allocate a significant portion of their research portfolios to basic research. It is basic research that will drive innovation. It is basic research that will provide the base of our future. But we don't know ahead of time how or in what direction. Furthermore, I believe that even countries with relatively small research budgets should provide significant support for pure basic research. Such research can be crucial in ensuring that the applied research, that the research that is focused in those particular areas that are most vitally important to individual countries and societies, basic research provides the basis for critical thinking, for experimental design, for creativity, and for establishing the highest level of science. One needs a combination of the basic research and the applied research. And in the context of biomedicine, only with support for basic research can applied biomedical research lead to the novel pharmaceuticals that will help improve world health and, as I said earlier, help us on the road to world peace. Now, there's another aspect of the relationship between science and government that I think is crucial in a broader sense to promoting the best government policies in a broad variety of spheres. And that is, government policies must be informed by good science. Science doesn't make policy, but science should inform policy. All too often, scientific data are ignored, distorted, or in some cases even fabricated to serve a political agenda. I think this is wrong. I think public policy should be based upon the soundest science available, and only in this way can the best decisions be made for a country and for a world. Now, unfortunately, many countries have ignored this principle, and I have to say that even my country, the United States, has at some times ignored this principle. Last week, I had an enormous honor. 1 week ago yesterday, I was in Washington, D.C. with President Obama. And we can see, are we on the screen there? We can see that at the White House last Monday, President Obama signed an executive order that had two impacts. First of all, he rescinded a political agenda-based rule that prohibited aspects of stem cell research in the United States. This was a rule that had been imposed that was not based upon science or scientific understanding, but rather was based upon politics. More generally, this rule exemplified a departure from the principle of using sound science to inform policy. And the second thing that President Obama did last week was to direct the White House staff to develop a strategy to restore scientific integrity to government decision-making. This order, I think, is extremely important. It's important for the U.S., and I believe it's important for the world. The message is that we all should use scientific knowledge in making the best decisions for policies, broad policies, policies concerning energy, policies concerning security, and policies concerning health. And in this way, we should all be able to make decisions that will lead us into a world with the best opportunities for world prosperity, for world health, and, I hope, for world peace. So with those remarks, I would like to stop. I want to thank, first, the many members of and visitors to my laboratory at MIT for the past 30-plus years, and also I want very much to thank you for being here to share my comments this morning. Thank you very much.
Thank you, Professor Horvitz, for the wonderful lecture. May we request Professor Horvitz to remain on stage. Professor Horvitz is an extraordinary researcher and has put in rare passion into his area of research. We hear that he was inspired to research this area after a close family member and another close friend of his passed away due to neurodegenerative disease. May I now invite Professor Muhammad Amin Jalaluddin, Deputy Vice Chancellor, Academic and International, to moderate the question and answer session. Professor Amin, please.
Menghadap Duli Yang Teramat Mulia Raja Muda Perak Darul Rizwan, Raja Dr. Nazrin Shah Tuanku Pro-Chancellor Universiti Malaya, ampun Tuanku. Patut mohon izin perkenan Tuanku untuk meneruskan sesi soal jawab seterusnya dalam bahasa Inggeris. Ladies and gentlemen, we have heard Professor Robert Horvitz delivered the last 45 minutes on the basic biomedical research drive, the discovery of new treatment for the human disease. Now, the session is open to the floor for question. Professor Horvitz are willing to accept your question and he respond to it. There are microphones in the hall on the left and the right and in the middle in the hall. Please identify yourself by names and the organization you come from. Yes, Professor Rauza, please come to the microphone there.
Assalamualaikum warahmatullahi wabarakatuh and good morning, sir. Can you relate actually the process of cell death to the physics of membrane?
To the physics of membrane?
Yes.
Not in great detail. The question, as you heard, was can I relate this process to the physics of the membrane? The membrane is the structure that surrounds each cell. And the major relationship is that as cells undergo programmed cell death, they are destroyed. And an integral part of that destruction is to destroy the cell membrane. Our bodies are very efficient, and so when cells die, the materials in those cells are not simply discarded, but they are reused. So the membranes are broken down and the molecules inside the cells are broken down and then reutilized by our bodies. This process is not the fundamental process of killing, but rather another step in programmed cell death, which I didn't discuss, that is involved in the destruction of the dying cells. It turns out we have also studied this step in detail in C. elegans, and what happens in us is very similar. There are implications for disease from our findings of these studies, but I think it would take me another lecture to describe that, and perhaps this is not the best time to do that in detail. Thank you.
Yes, please. Can you identify your name?
Good morning, Professor Horvitz. My name is Suzainor. I'm from the Faculty of Medicine, University of Malaya. I was just wondering, as you mentioned earlier, that your research, it started off as something really obscure, something really new, and people told you not to do it. But the thing is, while you were doing your research, you must have developed some kind of innovation that helped in the experiments as well, because for most of us, I think we are end users. We devise our experiments based on what is available for sale. For example, the microscopes that we use, the other things that we use to measure the cells' reactions and so on. What kind of innovation did you develop in your lab in order to help drive your experiments? Thank you.
Yes, thank you. There really are two answers to that. One, although we had no idea when we started whether there would be practical import to our studies, I was very aware that what we were studying was a fundamental problem for which there was no current understanding. And one of the lessons I learned from my PhD advisor, who was James D. Watson of Watson-Crick double helix fame, what Jim taught me when I was a graduate student was it's equally hard to study a problem that's unimportant as to study a problem that's important. So I knew that the biology was unexplored. So I knew we would learn something new. I just didn't know if it would be generalizable. Then in terms of the technology that we used, of course there was innovation and new technology added in many ways. One of the examples of technology that came into these studies, I think, exemplifies something very important in modern biomedical research, and that is the classic disciplines as labeled by many of us, as labeled by many departments, today are coming together. So it's very difficult to say that there are lines that should be drawn between biology and medicine and chemistry and computer science, mathematics, and even physics, because each of these disciplines is crucially important to help aspects of what goes on in the other. So that, for example, one of the technologies that we helped develop for our studies was the use of lasers in biological experimentation. And we were able to use laser microsurgery in this little worm to kill individual cells. And so if we killed one cell, we could then ask what happened to other cells in the animal, and that told us something about how cells interacted and how cells talk to each other. And so we used this technique from physics. We adapted it so that we could use it for our particular application, and this technique has been extremely useful to us, both in the studies that I outlined and also in many of the other studies that we've done. So technological innovation as well as biological innovation has been very important in the work that we've done. Thank you.
Please, sir. Yes, please.
I am Francis Achike. I am a professor of pharmacology from the International Medical University. Professor Horvitz, I thank you very much for a very stimulating lecture. As a basic scientist myself, I could relate to your description of what I perceive as the nothingness of basic research at the very early stages, and in your case, developing later to applied, applicable results in this case, treatment of several diseases. What I am trying to now get across in my question is this: how did you, from a state of what looked like nothingness, grow and progress into what is now a Nobel Prize project? And particularly, I want to ask, is there a role for spirituality? Or what is the role for spirituality in research? And in your particular case, how, if at all, there was spirituality driving you through these years? Thank you.
Okay, so the first part of the question is easier to answer than the second part. With respect to how I progressed and how those in my laboratory worked with me to progress, basically science proceeds a step at a time. And the most important thing, I think, is to do two things in parallel. One is to continue to be sure that one is asking important questions, and the second is to be careful that one obtains rigorous experimental data and makes conclusions that are driven by experiments that can be critically interpreted. And if one explores science, if one explores nature, and does so rigorously, frankly, nature is interesting. And if you do rigorous science, you will learn something new, and if you are focused on important problems, you will learn something new about important problems. As one does science, as I'm sure you are aware, one gets answers at one level, but probably each answer raises more questions than you had before you started. That is both the challenge and the excitement of scientific research. Spirituality is something that I think is very individual. I think it varies from person to person, how one regards spirituality and how one integrates spirituality with one's life, both personally and professionally. And I don't know any way to generalize about that. Some of the scientists I have known have been exceedingly spiritual in many aspects of their life, including the way they think about science, and some of the others, I would say, have been less so. But I don't know a generalization that I can make about the intersection between spirituality and science any more than I feel I could make a generalization about the intersection between spirituality and any single individual.
What about you?
What about me?
Yeah.
I would say my spirituality is more in the aesthetic level than, for example, on a religious level.
Thank you.
Please, Dr. Hashim.
Hashim Yaqub, I would like to ask you a burning question. There are very many types of university rankings in the world today. One of them is the Times Higher Education Quarterly University Rankings. I presume that you have heard of it. Some people are with the opinion that it is so important to get ranked by this THEQS university ranking. Have you heard of it? And what do you think of it? Should we worry too much about what the THEQS World University Ranking say about our universities all over the world? Thank you.
Okay. This also, I would say, is a complicated question, and the answer is probably yes and no. The external rankings, be they of universities or of scientists or of individuals in any walk of life, are complicated because they do provide an element of external validation, while at the same time many such organizations, and I don't know enough about this one to make any specific comments, many such organizations tend to be rather conservative. So they look towards really past achievements in the context of what has been done and what has been known, rather than looking and really evaluating future potentials. So, for example, in judging science, some of the most innovative science has been rejected by some of the most visible scientific journals. Why? Because innovative science is not popular, because nobody is paying attention to it. And it's only some years later, after it's been established, that the community recognizes that what was done was fundamentally important and truly pioneering. I think the same possibility exists with organizations of this sort. They will obtain criteria, but the criteria may not be the most forward-looking. On the other hand, there is the external validation that can be very important in drawing faculty, in drawing students, and in recognizing accomplishments. So I think one wants to basically be very conscious of these issues and not compromise internal standards and internal goals, but at the same time be sufficiently pragmatic to not ignore external agencies of this sort. And again, I say that with limited knowledge of this particular group, but just as a general sense of external validation as a route to quote, success.
Thank you very much, Professor.
Thank you. Yes, please. There are two. Sarah, can we give to the ladies first, please? Yeah.
Okay. Well, good morning. I am Dr. Gisela Gonzalez from the Center of Molecular Immunology in Havana, Cuba. First of all, I want to thank very much to Dr. Horvitz for being one of these persons that have spent his own life or his life in working for the benefit of the people. And then I want to ask Professor Horvitz some opinion about our work. In this case, I am going to refer to our personal work in our center, our Institute of Research in Havana. I will want to hear his opinion in some part of this work. We have spent a lot of years in working for new therapies targeting cancer. I mean, we have, in this case, we have approach of targeting the epidermal growth factor receptor system. That means we are trying to target the cell proliferation quality of cancer cells. In this case, as you have referred, cancer is one of these diseases where the apoptosis do not play its role, and then this comes together with uncontrolled growth. In our case, we have been working for several years in the development of a vaccine that targets the epidermal growth factor, that is the ligand. And then this vaccine provokes an anti-EGF antibody response, and this antibody blocks the binding between the EGF and the receptors, and then in some way stop the proliferation of these cells. This has been our work for several years. By now we have concluded phase 2 and phase 3 trial, and we have demonstrated that there is an increase in survival for patients that are being vaccinated when compared with the randomized control that do not receive vaccination. In fact, already now we have registered this vaccine in our country, and is available for all advanced lung cancer patients. My question is the following. What we always expected from this vaccine or from this kind of therapy is to have like a stop in growth, and because of this, like a prolonged survival. In fact, we have demonstrated that. We have, I'm sorry, we have prolonged survivals in patients that receive the vaccine, but not when compared with patients that does not receive the vaccine. But we have also observed several cases. I can mention around 10 patients in a population of 700 patients that we have already with this vaccine. About 10 patients that have not only stopped proliferation, but have a tumor regression. That means in some moment their apoptosis begin to work. And how we always have to try some explanation for this, because we are not only stopping proliferation, we are provoking cell death with this kind of vaccine. And then we always thought that in some way there should be like a kind of equilibrium that among apoptosis and cell growth, and that in cancer what happens is that the cell growth is more than apoptosis, and because of this, well, the tumor grows. And in this case, as we stop cell growth, the mechanisms go towards the apoptosis again. But well, this is just an hypothesis. We have never been able to test that. And I would like to know your consideration about this. What do you think about this? Why we are getting not only prolonged survival, but also disease, tumor regression, tumor remission, disappearance of tumor. That means that apoptosis is working there again. I would like to hear about your comments about this. Okay. Thank you.
Okay, I'm not sure I heard all of the specifics there, because there was some resonance with the microphone. But I gather the question, at least in part, is with this antibody that's been developed, you find that you can both block cell death and stimulate apoptotic programmed cell death. And in this way, you're actually intervening with the two sides of cancer, because as I described, cancer on the one hand involves too much cell division, and on the other hand, too little cell death. So if you can block division and amplify death, you're taking advantage of both worlds. Now, there are certainly data in the field, not from my laboratory, that indicate that there are particular human cancer genes, like the gene MYC, that is involved in both the proliferation and in the cell death. And so if one alters basically the control appropriately in cells, you can change from what would be a driving towards cell division to a inhibition of cell death. So I think what is known about the biology of cancer would fit very well with what you've just described, but I don't know if this particular gene would necessarily be the one that's important. The idea of using antibodies to intervene in this way is, I think, one of the most exciting areas of modern biomedicine, because it used to be, not very many years ago, that all of the drugs that were dealt with were very small molecules that were identified by pharmaceutical companies that were very much chemistry companies. And what has evolved over the past 20, 25 years with the emergence of genetic engineering and biotechnology is that there are now drugs that are so-called biologics, not little molecules, but much bigger molecules, and molecules that actually can be made using DNA as the basis of the synthesis of the molecules. For example, proteins such as antibodies or growth factors or other proteins. And the advent of biologics in clinical medicine has offered a whole new world towards therapeutics. So I find the area very important and very exciting. And of course, if one thinks about what's going on commercially, also very timely, as you may have noticed, that the leading, or at least one of the leading biotechnology companies, Genentech, has just been acquired by one of the major pharmaceutical companies, Roche, because Roche has recognized and has been interested for some years that this whole area of biologics is much the future of human drugs.
Thank you very much, Professor.
Thank you. Okay, we may have two more questions. We give to Professor Tengku Sara and Professor Lulaiming. Please.
It was a great honor to listen to your very clear and also very inspiring talk, Professor Horvitz. I am from the medical faculty, orthopedic surgery actually, so we are very, very applied really. And I would like to ask you, firstly, how or should we be getting into basic research, or should we leave it to those who are very inspired, etc., rather than the medical doctors in applied research? Actually, it's very attractive because I wouldn't agree that it's nothing, because impact factor, etc., actually favors basic science quite strongly. So we would like to get into this, but maybe you can give us some advice. And secondly, we are also probably in this country a little bit too fond of jumping on the bandwagon. So for instance, nanotechnology, stem cells, etc., when it comes in, we try to get involved, but sometimes a little bit too late. And maybe you can advise us how to start afresh and create something which is new on our own. Thank you.
Thank you.
Okay, so the first question is a question that I think can be asked at many different levels. Individually, for a researcher departmentally for a department, institutionally for an academic or hospital institution, and nationally. I don't think it is by any means necessary for every individual to do basic research. In fact, I think that would be quite counterproductive. And there are only a few individuals who have both the expertise and the talent to do both the basic research and the applied research. So I'm thinking more generally at the level of departments, institutions, and countries to have the interface, but one wants there to be communication so that those people who are doing the more basic research are interfacing with those people who have the more applied expertise, so that they're not acting essentially in vacuums, but rather are talking, interacting in a synergistic way so that advantages can be taken of both groups and to do something that is innovative. So that would be my sense of the way to best approach this. How to try to innovate within a country, or even, again, within a department or an institution or individually, is a very challenging question. And again, I think one wants to watch what's going on in the field and to be very aware of it, but also to not be afraid of doing something new, not be afraid of doing something that hasn't yet been validated by others. It's very hard to know ahead of time what's going to work, but as one says in finance, there is a risk-reward relationship, and some part of an investment portfolio in biomedicine should involve risky endeavors that have the potential to do something that is fundamentally breakthrough. Again, you don't want to do only risky endeavors, and you do want to be very aware of what's going on elsewhere, because there may be opportunities to contribute in a very important way to these emerging areas of science and in particular of biomedicine. But there may be other areas where you think about the problems that are facing the country, and you can think about novel ways of approaching those problems, but you don't know if those novel ways are going to work. And some of the investment should be in that area. So it's really, how does one promote creativity and innovation? And I would say there are two things to keep in mind, and actually a committee that I served on recently in the United States for the American Academy of Arts and Sciences, looking at innovation in our country, has come up with two specific recommendations. The first is support young people. Young people tend to be the most creative and the most innovative, and in many cases, in the U.S. and around the world, have had the most difficulty getting started. So by the time they're established enough to really do something new, they may be past their innovative prime. So develop programs that specifically support young people to do creative, innovative research. That's one thing that I would argue. The second is to really think specifically about high-risk, high-reward research programs in ways that are separate from standard research, and say a proportion of a research portfolio should be defined specifically for these high-risk programs, and then create an infrastructure, probably governmental, that would say we are going to seek, identify, and support some amount of high-risk research. And I think that combination of a conscious focus on young scientists coupled with a conscious focus on high-reward, high-risk, innovative research can help a country try to do things that are truly innovative, particularly in the context of the interests of that specific country.
Prof. Horvitz, I think we have one last question for the day. Professor Lulaiming.
Thank you. Thank you, Professor Horvitz, for a very delightful and inspiring lecture. I'm a professor of pathology at the University of Malaya, a fellow of the Academy of Sciences, Malaysia, and also I chair the Medical Ethics Committee at the University of Malaya Medical Center. I'll be very interested and grateful to know your views on the biomedical considerations for very basic research. For example, what considerations do we consider when we're looking at research on single cells like a yeast or a simple worm like a C. elegans? What about other single cells like stem cells and cancer cell lineages and cell lines? Where do we draw the line? And who are the we who draw the line?
Let me ask for clarification. What kind of line are we talking about? An ethical line?
Bioethical, yes. For biomedical research, what are the ethical considerations?
Again, a very complicated question. And so I served on another committee, and it was a committee set up by the U.S. National Academy of Sciences. And the goal of this committee was to establish ethical, practical, and forward-reaching guidelines for stem cell research, because at least in the U.S., stem cell research using human embryonic stem cells has been one of the most controversial areas. And there are many ethical questions that have been raised. And the answer to your question is in part specific. There are ways that one can think about the ethical issues and make some specific suggestions, but more generally than that, what we have suggested is that there be an ongoing group of people drawn broadly at two levels, both at the national level and at the institutional level for institutions that are doing such work, with people who are scientists, who come from the lay public, who have various religious backgrounds, who have political backgrounds, to think about, discuss all of these various issues on an ongoing basis as the science evolves, as our experience evolves, as our general knowledge evolves, to continually consider what we should draw as lines. I can give you personal opinions in detail about what I think in the context of stem cell research or research with other single cells, but all of these guidelines which I helped put together are available. You can find them on the internet, and I think what they are is current guidelines within the context of this more general recommendation: do not go forward blindly. Think about the issues and have a sufficiently diverse, educated, and open-minded set of groups to continue the dialogue at all times. Because what may seem right today, maybe will change tomorrow as we have more information and more experience. So I know that doesn't precisely answer your question, but your question is, in fact, one that many people have spent much, much time considering. It is not a simple question, and I cannot give you a one-sentence answer that would accurately depict what I believe to be the right answer to that.
Thank you.
Okay, thank you. I think, ladies and gentlemen, I think time is up, and the good things always some ends. Thank you for your active participation. Also, can you put together our hand to give a round of big applause to the Professor Horvitz.
Thank you, Professor Horvitz and Professor Amin, for the wonderful question and answer session. Could Professor Horvitz remain on stage again? We now take great pleasure in inviting Tan Sri Dato' Arshad Ayub to present a memento as a token of appreciation to Professor Horvitz. Tan Sri, please. Thank you, Tan Sri, and thank you, Professor Horvitz. We are deeply honored by the gracious presence of His Royal Highness, the Crown Prince of Perak Darul Ridwan. Ampun tuanku, bagi pihak hadirin sekalian, patut sekali lagi menjunjung setinggi-tinggi kasih di atas limpah perkenaan tuanku berangkat mendirikan majlis pada pagi ini. Mengumumkan keberangkatan Duli Yang Teramat Mulia Raja Muda Perak Darul Ridwan, Raja Dr. Nazrin Shah, Tuanku Pro-Chancellor Universiti Malaya, diiringi oleh pengurusi Lembaga Pengarah Universiti dan Naib Chancellor Universiti Malaya. Para hadirin dijemput untuk jamuan ringan di luar Dewan. Terima kasih.